Hello and welcome. Thank you for joining us today for the Sight and Sound Bites webinar. Today's topic in ophthalmology is changing cells using optogenetics to restore vision. So as you know, the Eye and Ear Foundation hosts these Sight and Sound Bites webinars each month, featuring different topics related to vision loss, hearing loss, head and neck cancer, sinus allergy, etc. I'm Carrie Fogel. I'm the Director of Development and the Foundation Relations at the Eye and Ear Foundation, and I'll be moderating today's webinar for everyone. A few quick housekeeping items before we get started with the presentation. We will not be using the chat function during the webinar today. Instead, we ask that you submit all questions into the Q&A box at the bottom of your screen at any point during the conversation, and I will submit those to the speakers following the presentation. We do ask that you keep your questions general about the content of the presentation and not about really specific personal questions that might not be relevant to the larger audience. And if there are any questions that you do have of that nature, we will send those directly to the speaker or the speakers, and answers will be sent directly to your email. And following today's presentation, there will also be a video of today's webinar posted online for you to review. So without further delay, I'd like to welcome our speakers. Today we have three speakers, Dr. Joseph Martel, Dr. Leah Byrne, and Dr. Jose-Alain Sahel. So I'll start with Dr. Martel. Dr. Martel is an assistant professor at the University of Pittsburgh, and he specializes in vitreoretinal surgery and diseases. Dr. Martel received his medical degree at the University of Nevada. He completed his residency training in ophthalmology at the University of California, San Francisco, and did his fellowship training in vitreoretinal surgery at Duke. His research interests include vision restoration therapies, retinal prosthetic devices, and robotic surgical tools for vitreoretinal surgery. Next, we have Dr. Leah Byrne, who is also an assistant professor of ophthalmology at the University of Pittsburgh School of Medicine. She received her PhD from the Helen Wills Neuroscience Institute at the University of California, Berkeley, and her lab develops gene therapies for retinal disease, among other activities. And her lab also engineers viral vectors with new abilities for improved capabilities to deliver therapeutic genes directly to the retina. And finally, of course, our esteemed chairman of the Department of Ophthalmology, Dr. Jose-Alain Sahel. Dr. Sahel is a clinician scientist conducting research on vision restoration and retinal degenerative diseases. He is a professor and chairman of the Department of Ophthalmology at the University of Pittsburgh Medical School and the Eye and Ear Foundation Endowed Chair and Professor of Ophthalmology at the Pierre and Marie Curie University at the Sorbonne. He is the founder and director of the Vision Institute in Paris, and has co-authored more than 500 peer-reviewed articles and holds more than 40 patents. He has received numerous prestigious awards and is the founder of several companies delivering ophthalmic therapies. So with that, I want to turn it over to, I believe, Dr. Martel will be getting us started. Again, please submit your questions at any point during the presentation, and we will do a Q&A session following the lecture. Thank you.
Thank you very much, Carrie. I want to welcome everyone, and I want to thank the Eye and Ear Foundation for putting this together. These are very informative for our patients, and I know that my patients in particular have mentioned to me that they really do enjoy these webinars and learn quite a bit from them. So I want to welcome everyone. Let's see if I can get these slides to work. Okay. So I think that over the next 30 minutes or so, what we'd like to accomplish is to first give you some context as to how the retina works and basic anatomy, how retinal degenerations affect the function of the retina, and attention to a subtype of retinal degeneration called retinitis pigmentosa. We'll then give an overview of various gene retinal therapy approaches. We'll discuss what is optogenetics, some of the basic principles on how it works, a novel clinical trial called Pioneer, which deploys optogenetics as a vision restoration tool, and then the future of optogenetics and some perspectives from our expert panel. So The eye is like a camera, but much more complex. The light coming into the eye is focused by various structures. You have the cornea, you have the lens, and it's ultimately captured and processed by a tissue deep inside the eye called the retina. The retina has various cellular components, components that capture the light, process it, and then send electrical signals along the optic nerve to the visual centers in the brain. So fundamentally, on the simplest way, basically what the retina does is it converts light energy into electrical signals that is then distributed as coded electrical signals to the vision centers in the brain. And so there are very special cellular components that comprise this retinal tissue. The light-sensitive cells in the retina called photoreceptors are distributed along the surface or along the retina as cones and rods. And those cells in turn capture the light and send signals to the nerve cells in the retina. And so there are basically two main types of photoreceptors, light-sensitive cells in the retina. There are cone photoreceptors, which are called cones because the shape of their outer segments is more conical. And those cells reside throughout the retina, but they're highly concentrated in the very center of the macula in an area called the fovea. Those macular cone photoreceptors are responsible for fine details in vision and color vision. In the more peripheral retina, we have a much higher concentration of cone photoreceptors. So our slender cone-shaped or rod-shaped photoreceptors that are in the peripheral retina that are more for lower light conditions, don't really give you much spatial visual acuity, but definitely help with visual perceptions. So if you've been a patient in our clinic and you've been on the retina clinics, you've probably gotten an optical coherence tomography imaging. And that's basically what this picture is. It's an image in cross-section of the various layers of the retina, and the different cellular components of the retina are distributed in very specific and organized ways. A light is shined into the eye. That light is then reflected back and gives us this pictorial image that allows us to take a look at the microstructure of the retina. And so when there's disease in the retina, things like macular degeneration, for example, here in the middle picture, there are areas of the retina that are degenerated. And there sometimes can be some scar tissue associated with that as well, which we see here. And that can impact the fine details in vision, color vision, and give a spot in the visual field. Normally, if it's the macula, it'll be in the very center. And then there are other types of retinal degenerations, like retinitis pigmentosa, for example, where the vision loss starts first peripherally, where there's constriction of the field of vision first. And toward the later stages of the degenerative process, there's loss of the macular photoreceptors and macular tissue, which causes a decline in not only the peripheral vision, but the center vision, and culminating into complete blindness in many patients with retinitis pigmentosa. And so the main issue with retinal degenerations is that unfortunately the retina does not have any regenerative capacity. So once that tissue has degenerated, unfortunately we don't know in 2022 how to regenerate that tissue. So depending on what the basis is for the retinal degeneration, there are many vision therapy approaches to try to improve the functioning of the patient. There are special lenses, like magnifiers, electronic devices, occupational therapy and training. The issue is that these devices don't necessarily improve the functionality of the retina. They just get the visual function to be optimized. And when you have very severe loss of vision, these types of therapies don't help much at all, if any. So this is an example of a patient with more end-stage retinitis pigmentosa. She only has the ability to detect whether or not the light is on or off. So she has severe visual impairment. And so those types of vision therapy strategies are not helpful in this type of patient. And ideally, in this type of patient, we would want to deploy a strategy that would have some vision restoration capability. And so with retinitis pigmentosa, it's a family of diseases where there's an inherited glitch in a specific gene that causes a more accelerated degeneration of the photoreceptors or the cells that help support the photoreceptors. There are many, many, many different genetic defects that can cause this type of problem. Again, it first starts out with loss of peripheral vision, but it's one of those diseases that can greatly impact patients' ability to see as it progresses. And so there's been a lot of research and interest in vision restoration approaches for these types of patients. Again, because the retina doesn't have regenerative capacity, once it's degenerated, we need to look at more alternative therapies to try to figure out, number one, can we bring back some vision either artificially or try to regenerate the retina? And those approaches include things like retinal prosthetic devices placed directly along the optic nerve or in the vision centers in the brain, retinal regenerative approaches. We've all heard about stem cells, but even transplantation of the eye that's been worked on. Those are sort of a little bit further off from being efficacious in patients, but certainly something that the scientific community is looking at. And then increasing in popularity and many different milestones recently are retinal gene therapy approaches, things like gene-independent approaches where we may know the specific genetic defect and be able to either replace that gene or help with the survival of the photoreceptors so that they don't degenerate. There's actually the first FDA-approved gene therapy treatment for a very specific type of retinitis pigmentosa that involves a gene defect in RPE65. And then there are more gene-independent therapies, things where it doesn't really matter what the underlying gene defect is, but we're deploying a gene therapy to try to revive or have some vision restoration capability in the remaining retina. And so I'll turn it over to Dr. Byrne to continue with some of the gene therapy approaches and some of the fundamentals of that.
Great, thank you, Dr. Martel. Hello, everyone, I'm Leah, and just as a reminder, I'm an assistant professor here at the University of Pittsburgh, and my lab is working on creating new gene therapies for retinal disease. So I hope just in a couple of slides to introduce you to the general concept of gene therapy and more or less how this works. So first of all, I'll try to answer the question, what is gene therapy? So it's basically a medical approach that treats disease by directly addressing an underlying genetic problem. So we're trying here to directly treat some sort of mutation rather than just addressing symptoms and complications. We're trying to directly address the underlying genetic reason for a disease. So we're actually, the way gene therapy works is we're using genes to treat disease. And so to do that, we need to transfer some sort of a therapeutic gene into the retina. So this slide is an illustration of the most common approach that we are using currently to transfer some sort of a therapeutic gene into the retina. So the way that that's most commonly done right now is actually using a virus. So the virus is just, it's a vehicle for carrying a gene into the retina. So we package a therapeutic gene into a virus, and then that virus is injected into the eye. After it's injected in the eye, the virus has a chance to infect cells in the retina. And then the gene that's packaged inside of the virus, it gets carried into the nucleus of a cell where that gene is then transcribed into some sort of a healthy protein that has some sort of a healthy action for the cell. So if we go to the next slide, please, you'll see. So I said that we're using viruses as the vehicle to carry therapeutic genes into the retina. And so viruses, for good reason, have sort of gotten a bad name over the past few years. So I just wanted to take a minute to address the fact that we're using viruses. Of course, viruses, they can cause some disease. And most of the viruses that you've heard of are ones that are associated with disease, like the virus that causes the cold, like common cold, or the virus associated with COVID. But the truth is actually that we coexist with viruses in the environment pretty well. And there are millions and millions and millions of viruses all around us all the time. And we coexist with those viruses normally very well. So a couple of interesting facts, for example, there's actually like 100 times more viruses in your body than there are human cells. So that's, I think, goes to show that viruses can be pretty neutral. Another sort of fun fact about viruses, there's like 140,000 different kinds of viruses in your healthy gut. So there's lots of viruses out there that are neutral or could even provide some sort of function. So the type of virus that is most commonly used for gene therapies right now is called adeno-associated virus, or AAV. There's many different viruses that have been investigated or that we're trying to use to transfer therapeutic genes into the retina, but AAV is sort of the leading virus. And you can see a picture of it here. So it's a very small virus. It's called a parvovirus, and there's not been any convincing evidence associating this virus with any type of disease so far. So it seems to be pretty non-pathogenic. And so the drawing at the top of this slide is actually a map of the genome of the virus. So it's a very small, simple virus. And like I said, it seems to be very safe. The other reason that we use this virus is that it has the ability to infect the cells in the retina. So Dr. Martel described how photoreceptors are the light-sensitive cells in the retina, and they're responsible for vision. So this virus, AAV, it is able to infect those photoreceptors pretty easily. And the other interesting thing that this virus can do, once it infects a cell, it delivers its cargo into the cell, and that cargo is the therapy. It can be a therapeutic gene in a gene therapy vector. And once it's delivered, that therapeutic gene is stable in the cell as long as the cell doesn't divide. So that is the reason that gene therapies could potentially be a one and done type of therapy. So with a single injection, in theory, it could be a cure that could last for the lifetime of a patient. And hopefully we'll get to that point. And the other reason we work with AAVs is that we can actually engineer new viruses. So this virus is malleable, and we can change the structure of it. When we change the structure of it, it will change the types of cells that it infects. And we can make different viruses, for example, that infect the retina more readily or more easily. And that's actually a lot of the work that my lab focuses on, is engineering new viruses with better abilities to infect retinal cells and more efficiently deliver gene therapies. So if we go to the next slide. I just wanted to give an overview of some of the different types of gene therapy approaches that we're exploring and trying to create. So there are different types of gene therapy approaches that are appropriate for different types of disease or different stages of disease. And so I'm just going to summarize a few of them here. So the most common type of a gene therapy approach is a gene replacement. So Dr. Martel explained how inherited retinal disease can be caused by mutations in a gene that encodes for some kind of a protein that the retina needs. So the most straightforward kind of gene therapy approach is just to replace that mutated gene with a healthy copy, and that's called gene replacement. And if you can click again, Dr. Martel. That is the most, so while gene replacement is sort of a straightforward approach, it's not appropriate for some types of diseases. For example, you could have a mutation in a part of your genome that is hard to access, or it doesn't actually code for a protein, it's doing some other kind of function in the body. Or it could be that you have a mutation that it doesn't just cause a loss of a protein, actually creates a bad protein that's doing something detrimental for your body. And so you can't just replace that with a healthy copy. You actually need to deal with the mutation in a different way. And so genome editing has become a really popular approach to dealing with those types of mutations. And what genome editing is basically just rewriting the mutation. So we would take out the broken strand, the strand that carries the mutation, and then rewrite it with a healthy copy. And then the third approach, if you can click, is what we're all here to discuss today, which is optogenetics. So in optogenetics, this is an approach that is really applicable in a case where the cells that you would like to treat have already died. And so if the cells have already died, you can't just replace a healthy copy of the gene because the cell is already gone. So optogenetics is a way to create artificial photoreceptors out of the cells in the retina that still remain, so that we can use a virus to deliver a gene encoding for a light-sensitive protein. And we can deliver that light-sensitive protein to other cells in the retina that still exist. So while your photoreceptors have already died and they are gone, we can create other photoreceptors out of remaining cells in the retina. So that's just an overview of different types of gene therapy approaches that will be applicable to different stages. And I believe I'll hand it over to Dr. Sahel at this point, who can explain a little bit more about those different stages and approaches.
Thank you, Dr. Byrne and Dr. Martel. So this schematic is representing the course of a disease that was already described by Dr. Martel and Dr. Byrne. Because of the mutations in retinitis pigmentosa, and because most of these are affecting rod photoreceptors, the cells that are enabling us to see in the dark, the initial symptoms of the disease is a loss of adaptation to darkness, difficulties at dawn or in dark environments. And this would be normally the ideal stage for a therapy because the photoreceptors are still alive, but they are non-functional. And by correcting the gene defect by gene replacement or gene editing, as was described by Dr. Byrne, you could potentially stop the disease and maybe in some cases improve the function in the cells that are still alive but are dysfunctional. Unfortunately, many patients are seen at later stages where most of the rod photoreceptors have degenerated and they have started to lose peripheral vision during the day with daylight. And we designed, my group, a therapy to try to protect the cone photoreceptors. Actually, Dr. Byrne, when she was in Berkeley, worked on that, and also when she spent time in Paris. And this is something which is going to enter clinical trials normally at the end of the year. The goal being to stop the loss of peripheral vision during the day, and more importantly, to preserve central vision for reading and face recognition. There is a stage where the cone photoreceptors have degenerated, initially losing the part that is responsive to light that we call the outer segment, which is a part of the photoreceptors that you see on the schematic. And once this part is lost, you still have the cell bodies that are there, but they are non-functional. There is another stage later, we put stage 4, where the cones have died, and only the innermost part of the retina, the cells that are connecting the retina to the brain, especially what we call the ganglion cells that are connecting the eye to the brain through the optic nerve, are still there. So the connectivity is still there, but there is no capture of light because the photoreceptors are degenerated. This is the stage where you can try optogenetics. If the cones are still there but non-functional, you can try to reactivate the cone. We call them dormant cones, and to reactivate the dormant cones. If the cones have died, then we decided to target the innermost cells, the ganglion cells, to try to reactivate them. What do I mean by that? If we go to the next slide, I'll try to explain what this means. What we try, and you see that in yellow, is to inject a vector, the virus, the modified virus that Dr. Byrne described, to target either the cone cells or intermediate cells in the retina, the bipolar cells, or the ganglion cells, the cells that are forming the optic nerve. What you have on this slide is just some of references on the work of my group on these aspects. So a lot of work that we started 12 years ago and showed that we could reactivate the dormant cones at that time. But we found out that this would be relevant only for 20% of the patients we see with advanced stages of the disease. This is why we decided to focus on reactivating the ganglion cells, the cells that are connecting the retina to the brain. And this is what you have on the right with a red square around it. So I'll describe a little bit the rationale and the preclinical work we did. The reason why we target ganglion cells is that there are many stages of a disease, and at all stages of a disease, including the later stage, which you see on the lower panel, the ganglion cells that are here painted in orange to visualize this are still there, which means that although the patient don't have any remaining vision, these cells are still there and they are still connected to the brain. So the goal is to reactivate these cells using optogenetics. How do we do that? We use a protein that exists in nature, in algae and some microorganisms, that was discovered like more than 25 years ago by a basic scientist working on these species. And they found that these species have the protein that is capturing the light and triggering an electrical response at the cell membrane that induces a response where the cells are able to move towards the light or away from the light. They have a cilium that enables that. This protein is very simple, and it does recapitulate a lot of functions that many proteins carry in the normal retina, in humans, in mammalians. But for that, we use 100 of proteins. So why do we use such a complex system if we could use a very simple system? Is because we are adapting to many levels of lighting, and it's a very sophisticated mechanism in the human retina. But sophisticated means also fragile. And so it's important to find a way to get a more robust way to capture the light. So the idea is to use these proteins from algae. The first research from several groups, including ours, was using the normal protein that exists in nature. But over time, many groups develop modified proteins that are more sensitive to light and safer to use. And this is what we did to move that into the clinic. Next slide. So we model that, and I showed you some of the many years of experiment we did in blind animals to try to reactivate the retina. What I'm showing you is a retina from an animal that was treated with this therapy, and we project onto the retina different shapes, like a square, a cross, or a circle, and each of these is eliciting a very specific response. What is a signature for each of the shapes. And what we could show is that the retina, what was blind, through the activation by these different shapes, is able to respond very specifically to that. Then if you move to the next slide, we developed a mathematical model to predict the type of vision that could be obtained potentially, and we compared that to the metrics we could gather from the experiment on the retina. And what we found out, and so it's a computational biology approach, using computational approaches to model, to record, to check. So it's a constant back and forth approach where you generate data, you build a model, you check the data back and forth, led us to show that potentially you could get a level of visual acuity that would be above legal blindness, which means above 20/200. So this was very strong motivation, which led us, with the help of a company that was formed out of a Vision Institute, GenSight, and a collaboration with my colleague Botan Roska in Basel, and many scientists at the Vision Institute, to start a clinical trial. The trial started in Paris, in London, and in Pittsburgh. And in Pittsburgh, Dr. Martel has been the principal investigator. So if you move to the next slide, it's describing the concept of a trial. So I'll just describe this slide and I'll hand over to Dr. Martel to describe the trial. This was called Pioneer, meant to target 9 patients with end-stage retinitis pigmentosa, which means blind from retinitis pigmentosa, no light perception or bare light perception, using a vector that is a modified vector, actually, that was developed by Dr. Byrne and Dr. Dalcara when they were at Berkeley. And this vector is a very strong vector to transduce, which means to express in the retina proteins. And we used a protein that was developed by a colleague at MIT, Dr. Ed Boyden, that is very sensitive to light in a spectrum of light that is safe in the red. The idea is to reactivate the ganglion cells that you see in yellow on this schematic, and then to send light onto the retina. And for that, we developed goggles that are capturing the light, responding pixel by pixel to changes of light, and to send the light onto the retina to reactivate the remaining ganglion cells. So now I hand over to Dr. Martel to describe the trial.
Thank you very much, Dr. Sahel. I feel very fortunate to be involved in this cutting-edge research and this inhuman clinical trial on optogenetics. So I'll advance the slide here. And so the main way that this treatment is administered is through an intravitreal injection. And that's a type of injection that we do quite commonly in ophthalmology. There are many different types of medications that we need to inject directly into the eye, into the vitreous, into the jelly, in order for it to access its target tissue deep inside the eye to the retina. So for example, in patients with wet macular degeneration or diabetic retinopathy, we commonly administer these what we call intravitreal injections. And this clinical trial is designed very similarly, where the gene therapy product is placed through an intravitreal injection into the jelly of the eye. It then goes to its target cells on the surface of the retina, the retinal ganglion cells. And once that genetically modified cell produces a light-sensitive protein, in this case, it's called crimson R. And that light-sensitive protein acts as a photo switch. And these light-sensitive proteins, as Dr. Sahel had mentioned, are very specific to a specific type wavelength of light, an amber light. And that's where the treatment is used in tandem with a pair of goggles worn by the patient to stimulate the genetically modified retina. This injection is performed in just one eye of the patient. It's the worst seeing eye. Patient undergoes many tests before the injection to determine suitability and also to determine which eye is worse. And so here's a quick movie of how these goggles work. In the front is a camera that takes pictures of the visual scene. The goggles are then equipped with a communicator to a pocket processor, a small computer about the size of a cell phone worn by the patient that has various algorithms to simplify the images in the visual environment, and then send a signal back to the glasses where an amber light is projected in a pattern on the genetically modified retinal cells. And so, oftentimes we're asked, why do the patients need to wear a special pair of goggles? And I think Dr. Sahel had alluded to the fact that, these are light sensors that are derived from algae, which is a fairly primitive organism. But those light sensors are fairly simple and robust. And the important thing is that they are directly stimulated by light, as opposed to more complex systems in mammals and in humans, where there's light that's captured by the opsin or by a molecule associated with the opsin. And then there's subsequent cascade of events that occurs in order for the electrical impulse to be propagated. The downside is that when you have this simple system, that it does require a little bit more of an intense light source. And in this case, it also has to be a very specific type of light, an amber colored light that's a very specific wavelength in order to selectively activate these cells that have been genetically modified in the retina. And so these are worn by the patient. The treatment is administered, and this is not a natural type of vision, it's an artificial type of vision. So there's multiple vision training sessions that occur after the treatment is administered with the patient wearing the goggles, and our very specialized vision training team here in Pittsburgh or at Moorfields if the patient's from the UK, or in Paris. And these patients undergo various vision training sessions so that we can make customized adjustments to the goggles if necessary, but also more importantly, so that the patient can actually learn how to use this artificial type of vision. And so the study is an early phase clinical trial. It's what we call a dose escalation study, where in early phase clinical trials, not only do we want to try to gauge efficacy, but first and foremost, we have to know if the treatment itself is safe. And so the way we go about doing that is that we give subsequent increases in dose of the gene therapy product, and reassessing at each stage whether or not the safety signals are reassuring enough to then move on to a higher dose. And so that's what's been done here. So far, there have been 3 cohorts of patients at various doses, and the last cohort of patients had the highest dose, which was deemed to be safe by a very special and sort of separate data safety monitoring board. And so currently we're at what we call the extension cohort, where we've determined the safest possible dose, which in this case was the highest dose, and are currently treating patients at this highest tolerated dose with the gene therapy product. So I'm going to show you a few videos. This is a patient undergoing visual rehabilitation after being treated. This is a patient in France, so there's some subtitles for you in case your French isn't too good. And you can see here that there's multiple objects on a table, and the patient's being asked to try to identify the location of the objects.
Parfait.
And preliminary results were quite remarkable. Safety signals were very good, as I mentioned before. This is an example of the patient so far with the best outcome, for which was reported in a Nature Medicine publication. It's a 58-year-old gentleman with end-stage retinitis pigmentosa, who before the study was only able to perceive light, no movements, no shapes, nothing other than just being able to discern whether or not the light was on or off. And after treatment and vision rehabilitation therapies, he was able to locate objects on a table with consistency, identify crosswalks. In addition, he was given a special type of cap with electrodes, what we call an EEG test, that allows us to correlate some of the visual perceptions occurring in the vision centers in the brain. And there was a correlation where the patient was able to identify the presence of objects and there to be the associated brain activity in the vision centers in the brain.
Premier sentiment quand j'ai effectivement eu le système sur les yeux et la première fois que ça a fonctionné, et quand je dis la première fois, ça a fonctionné dès la première fois. C'est assez impressionnant. À un moment, j'ai souvenir, dans une des expériences qu'on a pu faire, où je me suis retrouvé dans la rue, à chercher les passages protégés. Et il s'avère quand même que j'ai trouvé les passages protégés alors que j'avais le device sur les yeux, et que je ne l'ai pas retrouvé quand on a refait l'essai juste après sans les lunettes. C'est assez impressionnant parce que ça a excité quelque chose dans l'œil. Je me suis dit non, je ne vois pas, c'est mon cerveau qui voit. Ça donne des images qui sont très très particulières quand je dis que je vois les choses scintiller, que la perception c'est de la lumière qui vibre.
Those experiences are quite exciting. I'm going to show you another video of one of our patients here in Pittsburgh. Christine, do you want to show that video? I'm going to stop sharing here for a moment.
In this UPMC Physician Journal feature, meet an international team of researchers who set a bold goal, curing blindness, and see a first-of-its-kind success story.
Yeah, thanks.
This patient has a progressive disease which destroyed the light-sensitive cells in his retinas.
I was formerly diagnosed in my 30s, and by the time I was my early 40s, I was totally blind.
It's called retinitis pigmentosa, or simply RP. It affects more than 2 million people worldwide, and there is no treatment. The disease leads to complete loss of sight in most patients.
I just couldn't live with that. I just thought this is just not acceptable. So I just thought we had a duty to try to understand better the disease and to try to develop something.
What Dr. Sahel and his team developed is a technique called optogenetic vision restoration, and it's the reason Dan Dunphy is able to see these objects in front of him today.
In the middle is when the intensity decreases.
They're manipulating cells and proteins with light using a novel approach that combines gene therapy with a medical device.
How do I explain the experience? It is as if you were wearing glasses and you smeared Vaseline on them. So if I ask you, what do you see? You would see contrasts and changes in light.
Preliminary results of the Pioneer study were first published in May 2021, offering the first evidence that such a technique could partially restore vision in humans with RP. This is a representation of images taken by the camera as it detects light intensity. An international research team continues the study today through a collaboration co-led by Dr. Sahel and Dr. Bertrand Ruska, the UPMC Eye Center, the Paris Vision Institute, and GenSight, a startup gene therapy company.
The idea was to transform any remaining cell in the retina into a photoreceptor by expressing these proteins.
The genes used to achieve this transformation are the same ones found in glowing algae in nature. The therapy is delivered through a simple intravitreal injection.
It's actually one of the advantages of this treatment approach is that it doesn't require an invasive surgery.
Dr. Joseph Martel is the principal investigator in the United States for the Pioneer clinical trial. The emphasis now is on testing safety as well as early signs of efficacy.
We think that just one injection is actually sufficient. Based on the preclinical studies in animal models, we think that it takes anywhere from 3 to 6 months for expression. Those genetically modified nerve cells in the retina have the capability of sensing a very specific wavelength of light.
That's where the medical device comes in. These special goggles are equipped with a camera.
But it's working pixel by pixel to detect any change in light.
As the device detects changes in the surroundings, it projects amber-colored light pulses onto the retina, activating the proteins.
So they're fairly close together.
The information is transferred through the optic nerve to the brain, where it is processed as a visible image, much like a movie projector sends an image onto a theater screen.
And then the patient has to learn how to interpret this form, this type of images, because this is not natural at all. And this is certainly the most important, how the rehabilitation process has to take place with the patient.
For that, Mr. Dunphy has been returning to the lab once a week for nearly a year.
There's going to be a target on the screen that's going to be brighter than the rest of the screen.
Some weeks ago, I would just be able to see that something's there. Gradually, I learned to detect, or I should say deduce, as I'm scanning left and right and up and down, if it's, for example, a vertical rectangle, or if it's same in all directions, then it's either going to be a square or a circle. So there was no sharp, oh, here it is finally. It was a gradual thing.
I'm going to do something here, just a little bit, okay?
Dr. William Smith specializes in low vision rehabilitation.
Visual function is kind of what we're measuring on the screens, what their visual acuity is, measurements of their color vision, contrast sensitivity, and then we look at how a patient integrates that into their daily activities, which is functional vision.
Boundaries are the easiest thing to pick up. I could see the door jam. So that's why when I was standing in the hall out there, I scanned a little bit and I said, oh, there's the door jam.
Success for the patient would be doing anything that they want to be able to do that they couldn't do before.
I think it's about there.
It is.
My success and eureka moment is things that I never thought would happen in my career. I know what your vision was before. I know what it is now. And that to me is pretty amazing.
Obviously, we are working on the next generation of goggles, and we're also working on the next generation of optogenetics.
Dr. Sahel says research to improve the device, the gene therapy, and its delivery mechanism will continue at an even more exciting pace when the new UPMC Mercy Pavilion opens in 2023. It will include vision facilities that will function as a real-life laboratory.
What keeps me motivated is very easy. It's seeing patients, telling them we don't give up, we try. It's not easy, but at least it makes sense.
I must admit, it's exciting when from week to week, there's a change. Even this week, oh, there it is. To me, that was satisfying.
Get news and free CME at upmcphysicianresources.com.
Okay. So, you can see that things are quite exciting. It wasn't that long ago that these types of therapies were deemed in the realm of science fiction. And so it's very exciting time for us as doctors, and certainly for the patients, that there are some treatment strategies out there that should and will have an effect on vision restoration. And so far, this optogenetic trial that we've been conducting, the safety signals have been quite reassuring, and the early efficacy signals have been exciting. We think that there's definitely promise for partial vision restoration with this therapy, and the trial is ongoing, and we look forward to the next steps. We are doing some other enhancements to the trial in the future. I mentioned before about trying to correlate the electrical activity in the vision centers in the brain with visual recovery after treatments. Also trying to, with very special imaging, we've got Dr. Rossi in his lab, and our collaborators in France and around the world, trying to develop ways if we can image on a microscopic level and detect these optogenetic proteins being expressed after treatment, and then certainly engineering advancements to the hardware and to the glasses. So there's much more to do, certainly an exciting time, and I'll bring back Dr. Sahel to discuss some of the things up and coming and newer avenues.
Yeah, I'll be brief because I've seen that there are several important questions. So we are trying to see whether this is better than visual prosthesis. As you may know, we are also involved, and Dr. Martel is in artificial retina. So how do we compare the two? Some things are common, like the goggles and the training, but how does it get better? Trying to optimize the proteins that are capturing the light. Do we need the goggles? We think we need that, and Dr. Martel explained why. How do we optimize signal processing? How do we mimic the way the human body and the human retina and brain are processing the image? How do we improve the transduction, the effect of gene therapy? And Dr. Byrne has developed beautiful vectors that can do much better than that. And the good thing is that we are not the only one. We were the first in the world, the first in every field to do that. But now there are many companies that were formed over the years that are competing with us. We still think that we have a good edge. And the most important is that at some point, we'll be able to provide to patients useful vision. And this is where we believe this milestone is leading us, but more to come in the coming years. So we are available, the three of us, to answer any questions. I think there are a few in the chat, so happy to start.
Thank you all so much. That was a fabulous presentation. And, I'll just speak personally to say that that is probably the third or fourth time that I've seen that video, and it continues to move me and make me really excited for the next generations of treatment in optogenetics. So you're right, we have a few questions that we have time for. Let me get my window open. It just closed on me, and we will use the remaining time that we have. We have one, not a question, but just a compliment that I think we ought to read. This is from one of our attendees, and they've said, I have had 10 eye surgeries thus far and expect more in the near future from Stevens-Johnson syndrome and toxic epidermal necrolysis. And I want to thank you for everything you do, and thank you for the hope that you give people who need it just like me. So I want to share that with you first. We have a couple of questions. Are optogenetics used in any other medical fields?
The vision is the first, and we knew that it's kind of natural because optogenetics is working with light, and it's kind of obvious that this would be the first. But there are other fields of especially neuroscience that are getting into that, especially Parkinson's disease. And there is also a planned project for hearing in Germany that should start probably 3 years from now. Optogenetics is also used by many teams across the world as a tool to understand how the brain is functioning. It's like now, even, in Pittsburgh, there are tens of laboratories that are using that as a tool when they work on pain, on hearing, on the processing of information. So, and there is also a range of application in psychiatry that is being considered. There is a world leader in that field in California at Stanford, Dr. Desserot, and he's working on that. It's not yet in the clinic, but probably one day it could come there. But vision is ahead of the game by probably several years, I would say.
Thank you. Another question. We saw as part of the presentation that you use various methods to determine the efficacy of this treatment. You use EEG to measure output, you use a camera, but I would like to know how you as researchers and clinicians can use what information you learn from these tests and how they can inform the next generations of the treatment. How does everything you learn kind of inform the direction that you move?
So I can start maybe, and Dr. Martel would continue. Actually, until you get into patients, there are many things you don't know yet. Well, we do as much as we can at what we call the preclinical stage to get there. But the interaction between the patient, you heard at the end of the movie about the rehabilitation process, what the patients are describing, what is working for them, the edge detection, is telling us what is meaningful. And because we are using an outside processing system with the cameras and the goggles, there is always the possibility to optimize the goggles and the pattern of stimulation. And actually, this is already happening. We have a new generation of goggles. We try also to combine natural vision for patients who may have some natural vision, which is not the case in this trial because they are blind, with the artificial vision that is being restored. We currently have a big project, and Dr. Byrne is part of that, to actually modify the way we project the image onto the retina, understanding better the projection of the information from the normal photoreceptors to the ganglion cells, and to translate that into the shape of images that we would project onto the retina in our blind patients. So this is a project we just start with funding from the Department of Defense to optimize the vision. And this should help us to, with especially very specific vectors, to make sure that we target only the cell type that we need and not every cell, to improve the processing of information. So this is the next generation, very ambitious, but we work on it.
Yeah, I'll also add that, I think that the patients that are involved in this clinical trial, they actually themselves contribute quite a bit to the scientific advancement of this technology. We learned a lot from the patients on the types of things that they can see, what they can't see. And that's part of the reason of doing this clinical trial, is to try to figure out what are the capabilities of the technology and how we can improve on it.
Yeah.
Next question is from one of our attendees. Am I correct in assuming that optogenetics cannot be used with optic nerve cells that are already dead?
Currently, yes, because you need the ganglion cells, the cells that are connecting the eye to the brain, to be functional. So we are working around that two ways. One, there is, as you know, the Fox Center in our department that is dedicated to optic nerve regeneration. So, and actually one of the scientists we recruited from that is using an approach with stem cells to be targeted to regenerate the optic nerve. And actually my team in Paris has worked with combining optogenetics and stem cells. We have done that for photoreceptors, so this could be applied as part of the Fox Center initiative. The other approach is to try to get directly to the brain. We started that years ago, actually, in my team in Paris to do that. Problem is that the light doesn't penetrate very deep into the brain. So we are now we developed a new technology, which is called sonogenetic, where instead of light, we use sensors to ultrasounds that could get deeper into the brain. This is the early stage. None of this is in clinical trials, but it's just trying to be creative to get around this difficulty that if you don't have a viable optic nerve, you can't make it work so far, but we don't give up on that.
Is optogenetics an option for glaucoma patients?
So the same question, actually, because of glaucoma is affecting the ganglion cells. So this is really the focus we have at the Fox Center, and trying to solve this issue.
Great. What is, I think we have time for two more questions. So I'll read this one and then I'll read a combination of two other questions. What is the highest level of useful vision you've achieved with a patient already with this treatment? And has that patient achieved enough vision to travel independently?
Well, maybe Yossi, you can.
Sure. Yeah. So the patient that we showed you from Paris that we interviewed, he so far is the best possible outcome. I think that he previously was only able to see light and dark, and now I haven't worked with him personally, but my understanding is that he's able to navigate his environment much better. He's able to see crosswalks and objects. Now, he certainly doesn't see any significant fine details in vision, and we're not at that stage, to be honest. I don't think that this capability at the present moment would deliver vision to see fine details, get behind the wheel of a car, or to read a book or anything like that. That's not yet at that stage. We hope that might be something in the future, but right now it's very basic levels of vision. And the design of the clinical trial in its current iteration is really just for patients with end-stage loss of vision. So patients that really don't see much of anything and are blind. It's not yet appropriate for patients with better vision.
Yeah, I know that the time is running, so I see one question that we have not. Someone is asking, do patients have to pay for a clinical trial? Never, just never. Anytime you hear that there is a trial where people have to pay, it's not a scientific trial. A patient should never have to pay anything for a clinical trial, just to be clear.
Thank you. Yes, that was going to be part of my next question. So you answered that no one will have to pay to participate in a clinical trial. And the other part of the question is, how does someone, participate in a clinical trial for optogenetics if they think they may be a candidate?
So we see the patients in the clinic. We have a center where we see patients with this type of conditions, specifically every week. So we are several, Dr. Martel, Dr. Rosin, and myself, and patients who are eligible for a gene therapy trial or optogenetic trial, we inform them when the trial is available, or because we know them, we have a database and we identify the patient. So Dr. Byrne is working currently on another gene for gene therapy. So we keep accumulating the information so that the day we are ready to start, we'll know a lot about the patient. There will be already aware that something is popping up in the future, and then we'll inform them. But then they have a lot of thinking to put into that, because it's quite of a commitment to get into a clinical trial. You have to come back often, you have to be tested, and it's exploratory. We don't know yet the answer.
Well, I want to thank all of you again. Thank you to our speakers, Dr. Martel, Dr. Byrne, Dr. Sahel, for taking time explaining this really exciting new therapy in restoring vision. And with that, I think we'll conclude. I want to remind everybody that a recording of this webinar will be available on the Eye and Ear Foundation website. And if you have any questions that you didn't think of during today's presentation, please feel free to email Craig Smith, and he will get them to our speakers and can get you an answer. So again, thank you. Happy Friday to everyone, and enjoy your weekends.